Hi everyone, this is a lesson on Punnett squares and solving Punnett squares. We're going to begin with monohybrid inheritance, or the inheritance of one trait. All right, so let's start with the parents. We have to determine the phenotype and genotype of each parent. So let's say that both of the parents, and we're going to take into account plants, just like the father of genetics, Gregor Mendel, did. Plants, in this example, are heterozygous for the trait of height. For this trait, a tall plant is an uppercase T, it's dominant, to a short plant, which is a lowercase t. All right, so here are, we have to take into account the parents. We've got parent 1, and parent 1 has a genotype of uppercase T, lowercase t, so two different forms of the gene, which we call heterozygous, times, we're going to cross it with parent 2. And parent 2 is also heterozygous for this trait. So here are the genotypes, uppercase and lowercase t. Uppercase T, lowercase t, both parents are heterozygous. So these are the parents. We're going to cross two parents, parent 1 and parent 2. We got to figure out the gametes. So we got to determine which alleles would be found in all of the possible gametes, egg or sperm. All right, so let's draw the gametes. We will, gametes, remember, are reproductive cells, and they include sperm or egg. They contain n number of chromosomes, or half the number. Okay, so let's take into account sperm first. So I'll draw 1, 2, and I'll draw the eggs without the mobility. All right, so we've got sperm and egg. And looking at the parents, each sperm is going to contain n number of chromosomes, or n number of traits. So this big T we can place right here, and that little t we can place right here. And the same goes for parent 2, eggs. Now this follows the law of segregation, which states that each gamete contains only one allele for each trait. So if you look at, we only have one trait here, and each of these gametes, sperm and egg, each contain one allele for each of the traits. So we've satisfied the law of segregation. All right, so now we have to draw the Punnett square. So we'll draw a table that is a 2 by 2, 2 by 2, and draw a table with enough squares to do our cross based on the number of different gametes that each parent can produce. All right, so let's draw our Punnett square, which is a box. And it's 2 by 2. And we're going to place parent 1 gametes here across the top. Big T, little t, those are the possibilities for that one trait. And we're going to place parent 2 gametes always here along the side. Uppercase T, lowercase t, there. So now we're going to write out the new genotypes. So we have to fill in the table by combining the gametes genotypes. So we're going to keep the alleles for the same trait together, and we're going to always write the dominant allele, or the uppercase allele, for a trait first. So we're going to cross parent 1 with parent 2, so we end up with a possibility of uppercase T, uppercase t. We're going to cross parent 1, lowercase t, with parent 2, uppercase t, but we're going to, regardless of which parent contains the dominant allele, we always write the dominant first. So we'll cross uppercase T from parent 1, lowercase t from parent 2, and then we'll cross lowercase t and lowercase t for our homozygous recessive alleles. All right, so here are all the possible genotypes. So let's write them out. So we have uppercase T, uppercase t, uppercase T, lowercase t, and lowercase t, lowercase t. Now, as a reminder, the two uppercase alleles, if they're the same form for the same trait, we call these homozygous dominant. Here we have heterozygous, forms of that gene. And here we have homozygous, but in this case they're two lowercase, so it's homozygous recessive. So those are the possible genotypes from a cross between two heterozygous parents. So this is a classical Mendelian monohybrid cross between two heterozygous parents. So let's take a look at what the results are. So you're gonna be asked to calculate percentages. And so in this case, we have how many, or what is the percent tall? What is the percent short for all of the possible outcomes, or for the outcomes of this cross, divided by the possible outcomes, multiplied by 100%. And then we have the genotypic ratios, in which case we will always use numbers and we will always write out the genotypes. Homozygous dominant compared to heterozygous, in this order, compared to homozygous recessive. So we'll keep those in that order. We'll also be asked to calculate the phenotypic ratios. So let's go ahead and calculate the percentages of tall offspring that are predicted to be an outcome from this parental cross between two heterozygous. Well, if we look, because this is a complete dominant trait, the uppercase T, or the dominant allele, will mask the recessive allele. So therefore, there are 1, 2, 3 chances of resulting in a tall offspring, versus 1 out of 4 possible chances for resulting in a short offspring. So we're going to figure out the percentages by dividing the outcome, 3, divided by the possible outcomes, 4, and then multiplying by 100%. So we end up with 75% chance that these offspring could end up tall, and a 1 out of 4 divided by, or times 100%, 25% chance that the offspring would be short. And then we'll look at the genotypic ratio, so we'll just count them. So it will be a classical 1 to 2 to 1 ratio for the cross between two heterozygous parents. And then resulting in these two genotypes will then go on to produce tall offspring, so that's 3 out of 4, to the chances of having short offspring, which is the physical trait, so it would be a 3 to 1 ratio for the phenotypic ratio. Okay, that's how we set up a monohybrid inheritance cross.